Behind every discovery, there is always an ancient dream. The scientists of today are breathing life into the myths of the past. Prometheus, punished by Zeus for giving men fire, was chained to a rock. A vulture comes every day to devour his liver, which reconstitutes itself every night, prolonging the sufferings of the Titan for eternity. Human regeneration, man's eternal dream. In reality, the liver is able to reconstitute itself almost entirely in 5 or 6 months, paralleling the myth. As all of us have experienced, the blood and the skin also partly possess this astonishing capacity of renewal. Recent advances in research have shown that the brain too has this property, but to a lesser extent. At the root of this potential for regeneration are cells, known as stem cells.
What you're looking at here is a culture of neural stem cells, and particularly you should focus your attention to these cells. What you can see here is a dark-phased nucleus and the surrounding cytoplasm. Now what the cell is going to do is to retract its processes, as you see here, to round up, and what the cell is trying to do now is to divide, and starting from this single cell, you should see 2 new cells emerging. Here they come, 1 and 2. Now the cells will flatten out, extend their processes, and become 2 new cells with their nuclei and cytoplasm. Come on, baby, do it. Okay? They work like dogs, for sure. To understand what a stem cell is and as to why we apply a certain definition to say that a given cell is a stem cell, we need to understand what the function of the stem cell is. A stem cell in a tissue is similar to the maintenance service, meaning we have a tissue, the tissue is made up of very small units, like a wall is made of bricks, a tissue is made of small cells. As soon as you are injured, there is a release of chemical signals in the tissue that trigger the stem cell to become activated and to generate a functional, mature progeny that goes into the tissue and repairs your wound. That's why the stem cell is there. On top of the stem cell being capable of putting new mature cells in the tissue all throughout life, even if there is no damage, but because your cells are being consumed physiologically.
A stem cell is above all a cell, and one of the major functions of every cell is to divide and give rise to 2 twin daughter cells, which will each in turn give rise to 2 daughter cells. So first there is 1 cell, then 2, then 4, then 8, and so on. If things were always as simple as that, the result would be a great mass of cells. However, sometimes the result is a baby mouse, a baby tiger, a baby ladybird, or a baby human. Why? Because not all the daughter cells have the same fate. But let's go back to the beginning, and even before. As soon as the egg is formed by the fusion of an ovule and a sperm, it rushes to divide again and again, until the embryo resembles a little blackberry composed of 8 stem cells, which are highly potent. Each of them could, in theory, give rise to a full embryo, a fetus, and finally a baby. Then, around the 4th day, the dividing stem cells begin to specialize. They give rise to 2 types of cell. In the center, a small internal mass will eventually become the future fetus, and at the periphery, the cells that will go on to form the placenta. The cells of the internal mass are called embryonic stem cells. Research on these cells only uses cells taken from embryos generated by in vitro fertilization, but not implanted in the mother's womb. Hundreds and thousands of such embryos are stored frozen all over the world. These embryos are known as spare embryos. If we take cells from the internal mass, we find that every one of them has the potential, on dividing, either to form cells identical to themselves, which can be grown to large quantities in culture, or, alternatively, to form muscle, cartilage, neurons, blood, and other tissues. As the embryo advances in its development to become first a fetus and then a baby, most stem cells gradually specialize and lose their ability to form a wide diversity of different types of cells. Nevertheless, the fetus is an important source of stem cells. Scientists obtain these fetal stem cells from the umbilical cord or from fetuses following elective abortion or miscarriage. Adults also have stem cells, albeit in small numbers. Most of these cells are more specialized and are normally able to replace only certain types of cells during the regeneration of a damaged tissue or organ. Adults are therefore another source of stem cells. In conclusion, we have stem cells from the embryo, which one day may be used to treat, for example, Alzheimer's disease, Parkinson's disease, diabetes, and heart diseases. We also have stem cells from umbilical cord blood, which are used today against leukemia, and tomorrow could be used to treat the same diseases as embryonic stem cells. And finally, we have stem cells from the adult, which are currently used to treat hematological diseases, and in the future could also be used to treat all these diseases.
This future is not so distant. Scientists have found the solution to an enigma that the Sphinx itself would never have thought of. How can you change the fate of a cell? The answer comes from basic research with adult stem cells taken from animals.
La plasticità delle cellule staminali.
We can define the plasticity of stem cells as the capacity to change fate when taken under experimental conditions from one tissue and put into another tissue. For example, you take stem cells from the bone marrow, put them in regenerating muscle, and they will form muscle cells. But the same cells are also capable of forming brain cells when you put them into the brain. In any case, the change of fate is strictly regulated by the environment.
The other important thing is that the cells actually reach functional maturity. We placed brain stem cells in contact with muscle cells, and what happened was that the brain stem cells turned into muscle cells. They actually turned into fibers, and they made new muscles. So they have completely reprogrammed their identity to go from brain to muscle and actually to functional muscle. This is done in a petri dish, and it's done in a way that allows you to quantify the phenomenon, to say how many brain stem cells turned into muscle, which is approximately 10% of the total stem cell population.
Extensive studies in animals are required before a treatment for humans can be developed. But, once again, myths can be made reality. There is already an example of medical treatment by stem cells which has been used for 10 years. Patients suffering from leukemia, a cancer of the white blood cells, can today be treated by the transplantation of blood stem cells obtained from the placenta. This is now simple and routine.
So, you're here to talk about donating placental blood. Once the baby is born, the midwife will collect the blood remaining in the umbilical cord. This blood was circulating in your baby. We therefore do not take blood either from your baby or from you. This blood contains stem cells, which are very young cells, with a strong potential for development. Indeed, they make it possible for platelets and white and red blood cells to develop in your baby, and therefore in the child who will receive them. So we're going to collect it into a small bag. There's about 80 milliliters on average, that's all.